To investigate these animal and human models, there are a number of non-invasive measures of brain activity. So we have invasive measures and non-invasive measures. So some of the ones that we talked about previously were invasive measures. If you were to do deep brain stimulation, that would be a very invasive measure. But we also have non-invasive measures that look at brain activity. So in particular, we have EEG. This is electroencephalography. This particularly tries to assess electrical activity in the brain. So this measures the summed graded potentials from thousands of neurons. So on the whole, whenever you have a group of neurons active, then these summed potentials, again, if these active neurons are active, then they have these variation in electrical charges that occur, and these summed potentials, these continual electrical charges and action potentials, can actually result in very weak electrical fields. And these very weak electrical fields can be assessed by EEG. You also have ERP, which is event-related potentials. This is basically EEG technology that is tied to a specific sensory event. So you could present someone with a stimulus on a computer screen, and then you would use EEG to see what brain regions are active. These more active brain regions would have more graded potentials, they would have more action potentials, and this would result in a slight electrical field that the EEG imaging device could pick up on. This is overall what it looked like. You have these electrodes that they sense the electrical field, and as the person perceives information or does particular tasks, then these will be really, really close to the scalp, and so these can only assess activity that is more near the cortical surface. And so these electrodes would be placed in particular regions, and then they would read the electrical activity, and this electrical activity could be converted to a color that would show sort of a representation of like, okay, this region is more active, and so on and so forth. So then you can see differences in these potentials as a function of time and activity and space in the brain. EEG has a very rapid response, so because neurons these graded potentials occur at a very rapid rate, then you have excellent timing with regards to assessing when neurons are active, but then the spatial information can be quite fuzzy, so it's not as exact in terms of spatial information. By spatial information, I mean where on the brain this activity is occurring. So if I were to give you an example here, if this particular brain region right here were active, then you have only a certain number of electrodes on your brain, and if it's right here in the middle, then all of these electrodes would be picking up on some of this. And so then you would have this larger array of activity that's actually coming from more sort of the smaller point. So the timing in terms of general area is really exact. As soon as something is presented, it can be registered, but then which brain regions are involved is a little bit harder to determine with EEG. And then you have magnetoencephalography. This is MEG. Similar to how neural activity creates an electrical field, consistent neural activity can also create a magnetic field. A magnetic field is basically electrical charges in a single direction. So neurons tend to line up in cortical gray matter and be in a particular direction. So as these neurons fire, then they will create a very small magnetic field. And this magnetic field can also be detected. And because this magnetic field has a spatial orientation to it, then the spatial resolution is better than EEG and ERP, but MEG is quite expensive. It has a sort of more expensive machine and field that can detect these very, very small changes in magnetic field as a function of neural activity. We also have a couple of static imaging techniques. This would be a little bit similar to X-ray, and how an X-ray just assesses or measures what is going on at a particular time point. So one of them is computerized tomography, and this is basically like an X-ray beam that's passed through the brain, and it's passed through at many different angles. And so through reconstructing all of these different angles, you can then reconstruct a 3D image. And so this is really good for when you want to get a snapshot of what is going on in a particular time. It does not offer you anything with regards to across time. CT scans are often used to assess if an individual has had a stroke, an aneurysm, because they are really sensitive to changes in tissue. And they're not that expensive because it's basic X-ray technology. But they also do not have as much flexibility as other imaging techniques, which I'll be talking about shortly. Here's an example of a CT scan, and so you can see here that they're assessing the CT scan after a person has had brain damage, and this is an example here of the lesion in their brain. And so it's dark because it is filled with cerebrospinal fluid, similar to the ventricles. And so this here you can see very distinctly that, oh, this person has damage to a particular region of their brain. Another very common static imaging technique is magnetic resonance imaging, known as MRI. MRI is probably one of the most well-known imaging techniques. It gives you excellent spatial resolution. This is a technique that produces a static three-dimensional brain image by passing a strong magnetic field through the brain, followed by a radio wave, and then it measures the radiation emitted from hydrogen atoms. And I'll be giving you a little bit of a video that you can watch later on in this chapter to learn more about MRI specifically, as well as functional MRI, which I'll be talking about when we get to dynamic imaging techniques. This is an example of an MRI picture, and this is actually a low strength field picture, so we actually have much better imaging technology nowadays for even creating even more crisper images of the structural differences within the brain. So this is cerebrospinal fluid. This here is brain tissue. Here you have the skull. And then the final static imaging technique is diffusion tensor imaging. And diffusion tensor imaging actually uses the MRI scanner. So one of the things that I was talking about with regards to an imaging technique that has lots of flexibility, computerized tomography or CT scans, they are really accurate for, again, a static image, but they don't have a whole lot of flexibility, whereas MRI scanners, they have a huge amount of flexibility in terms of the fact that they can assess structural, the sort of static brain image, they can assess functional, which I'll be talking about shortly, and they can assess this different form of functional known as diffusion tensor imaging. This detects the directional movements of water molecules to image nerve fiber pathways in the brain, and this is also used to delineate abnormalities in the neural pathways. So to give you an example of what a DTI scan looks like, a DTI scan primarily focuses on white matter pathways, and so water molecules, when they are a part of cells, in particular when they are a part of axons. Axons are very thin and they're very small, so the water molecules have a particular shape, and that particular shape, then when you try to detect the directional movements of water, you can detect that particular shape. So you look for sort of this long, narrow sort of nerve fiber pathway, and you can try to find the direction of the sort of small tubular water molecules. And in finding the sort of direction along which axis it sits, then you can map out these white matter tracks or these white matter fibers throughout the brain. So these colors are coded according to direction. And so here the red is again coded for the sort of this direction, this lateral view. And then you have the green that is sort of this direction, up and down. You have yellow, which is sort of more diagonal, and then you have blue, which you can't actually see here, which would actually be sort of three-dimensional. It would kind of go up higher into the brain, sort of lower into the brain. And next we have dynamic brain imaging techniques. Okay, so the most well-known of these is functional MRI, and this assesses the changes in oxygen content of the blood, how these alter the magnetic properties of water. And here is a video which we can use to watch this. I'll also post this online because this video is about 5 minutes long, so I'm not going to actually include it as part of the lecture. So you can pause this lecture and watch this posted video, or you can keep on watching the rest of this lecture and then watch this video afterwards. But it's really important to watch this video because the video describes a little bit about how MRI works. And MRI is, it's really the imaging technique that basically has made it a little mainstream, that it comes up in movies and it has, you can look at what's going on in people's brains using MRI, and so it's kind of neat to know a little bit more about how it works. The next dynamic brain imaging technique is positron emission tomography, also known as PET. And you can see all of these have these abbreviations, the anachronisms, because no one wants to say functional magnetic resonance imaging all the time or positron emission tomography, so they all have the short forms, fMRI or PET. So PET is an imaging technique that detects changes in blood flow by measuring changes in the uptake of compounds such as oxygen or glucose. And it's used to analyze metabolic activity of neurons. And what you do as part of PET is you actually have these radioactive molecules that are injected into your bloodstream. And this sounds like a really big deal, and PET is actually considered to be a little bit of an invasive imaging procedure. However, these radioactive molecules are harmless for your body. So all it is these molecules that radioactive doesn't mean that they have this sort of harmful radioactivity. They actually have a real rapid radioactivity that when the molecules enter the bloodstream, they attach to oxygen or glucose so they can travel to the active regions of the brain, and then they start to decay and decompose. When they decay, then they split, and the detection of these decaying radioactive molecules can be detected. And you can tell a lot more about what is going on in the brain by where these radioactive molecules are detected. So here, some of the advantages of PET is you can actually detect the decay of hundreds of radiochemicals, and this allows mapping of a wide range of brain changes and conditions. And so some of these radioactive compounds can be used for stroke, so you can actually assess where blood flow is going on in the brain. So if an individual has a small pooling of blood flow, if there's a clot, because then if there's a clot, then oftentimes oxygen and hemoglobin will cluster around a clot that can be blocking blood flow. And so then PET can be really advantageous for identifying that, because then if the oxygen and hemoglobin molecules are trapped there, then they'll spend a little bit more time there, and in that time passes, some of those molecules will decay and the machinery will detect it. So there's certain things that you can do with PET that you cannot do with MRI. Similarly, PET can also be used to study the density of receptors, metabolic activities. You can have very tailored compounds that are injected into your body to try to target or go to particular parts of the body to see how those are functioning. And it's also widely used to study cognitive function. Here's an example of what a PET scanner looks like. It kind of looks a little bit big and scary, but it's not actually. A small amount of radioactively labeled water is injected into a subject, and what happens is active areas of the brain, they're going to use more blood, and just as also active areas of your body, muscles and stuff like that are a little bit more active, they will use more blood, and thus more of the radioactive labeled material will travel there. Once it travels there, you have these photon detectors. So these annihilation photon detectors, they detect these annihilation photons. So what happens is that positrons, from the radioactive materials are released, and they collide with electrons in the brain. When they collide with electrons in the brain, a photon, which is a form of energy, is produced, and it exits the head. So we're not necessarily aware of it, but we actually do have really small molecules that are entering and exiting our head at any one time. And so the PET detects this PET scanner detects the exit of these photons. And when these photons are released, they are released at exact 100-degree angles. So if a photon is detected over here, then it also means that a 180-degree one was released over here, and so they can then use these 180-degree mapping, to pinpoint particular regions of the brain. And so it sounds a little scary to have this radioactive compound injected into your body, but they really do nothing. They just basically are detectors that are released. Now, I've talked a fair amount about the gene-environment interactions in previous chapters, but I'm going to cover it again here because it's a particular part of this chapter. So I just want to make sure that I'm talking about all of the techniques that are used to assess the brain and behavior relationship. But given the fact that we have talked about it in prior chapters, I'm not going to be spending a whole lot of time talking about it because we also will continue to talk about it. The gene-environment interaction is something that is a relatively continuous theme throughout this course. So we have alleles that come in these specific forms, and these allele pairs are genes, and they have individual characteristics, and they are linked to behaviors. So the gene-environment interaction can be when particular events, such as chronic stress, traumatic events, drugs, culture, disease, general cumulative experience, all of these, both points A and B, can affect how genes work, and they can also affect how particular genes are expressed. So to give you an example of how this would occur, when our body is chronically stressed, then there are particular compounds that are released, some hormones, in particular cortisol, and if you have continual cortisol release, this signals to your body that you are in a stage of chronic stress. And if you are in a stage of chronic stress, then your body thinks that it needs to conserve energy. Thinking back to earlier, more quote-unquote animalistic times, when earlier in our human or hominid development, the times of stress basically would have been considered times of famine or disease. And so what would have been the most adaptive response to our body in these times of famine or disease-related stressors would be to try to conserve energy. And by trying to conserve energy, energy in your body is stored as fat. So what would happen then is that chronic stress can cause the activation of particular genes that do one or multiple things. One, it can cause you to seek out foods that would be rich in fat and calories, because if you are undergoing famine, then you want to be automatically orienting towards food that would be able to sustain you. The other thing it would do is it would put your body in a state of trying to conserve those calories and that energy. So it would put your body in a state of trying to conserve and make fat. And so this is a prime example of how the body's response to chronic stress that at one point would have been adaptive is now, in the present environment, maladaptive, because we generally have so much chronic stress in our lives, but we also do not have any sort of situation where we have any sort of scarcity. Rather, we have the opposite. Instead of scarcity, we have everything that we could possibly need at our fingertips. So when our body is in this chronic stress state, then what it does is it tends to crave carbohydrate and fat-rich foods, and it also tends to conserve those eaten calories as fat, which of course in the present environment actually leads to unhealthy behaviors and unhealthy physical outcomes. We also have epigenetics. So epigenetics measures gene expression, and so the example that I just gave would have some epigenetic factors because stress can cause changes in gene expression. So epigenetics is also investigated by examining identical twins, and to show the difference, to show the wide impact that environment can have, there have been identical twin studies. And what is interesting is that despite the fact that these individuals, they are exactly the same, by the time they are age 50, their gene expression is as different as regular siblings. Most of you have siblings. You know how different you are from your sibling. So to take individuals that were born genetically identical, and then after 50 years, their gene expression is as different as siblings, really shows how impactful the environment can be on gene expression. What is also not known is that gene expression changes as a consequence of the environment, drugs, culture, toxins, that some of these things can actually be passed across generations. And so they can cause permanent changes permanent genetic changes that would be passed to offspring. And this here is a summary table of how we examine the brain. So most of these things are things that I've already talked about. So again, linking brain and behavior. This reviews, we have tissue analysis, behavioral analysis, the brain perturbation that refers to lesions in the brain, making lesions in the brain, or stimulation. So again, for humans, you can make lesions to remove a tumor, or you can have deep brain stimulation for testing. Optogenetics refers to using light to activate specific ion channels, and this is related to behavior. This is relatively new technology. Then you have measuring electrical activity. So we have, again, recording graded potentials. This is EEG, ERP, or MEG. And then if you were to record individual action potentials, this would be single cell recording. This is typically done in animals, although there are very specific times, such as when I described earlier in the lecture about how you can have electrodes inserted in your brain to prepare you for surgery, that you would also be doing single cell or multi-cell recording in humans. And then you have multiple brain imaging techniques, static images, CT scans, MRI, or DTI. Dynamic images are going to be fMRI, PET, or functional NIRS, and neuroinfrared spectroscopy, I believe is what this means. And you can learn a little bit more about this in the textbook. I didn't talk about it too much in this lecture. And then you also have these chemical and genetic measures. And this is some of the stuff that I was just talking about, how analyzing particular genes that people have and relating that to behavior, and then epigenetics, identifying the effects of experience that people have in the environment on gene expression and the brain and behavior relationship.